Hello, good day everyone. I'm Celine Yong, Kuan, and today I'll be presenting on the antibiotic gentamicin. To begin with a brief history, the gentamicin was discovered in the 1963 from a soil bacterium called Micromonospora purpurea by Dr. Marvin Winston and his team. It is produced through microbial fermentation, making it a classic microbial natural product derived from microorganisms through bioprospecting. It belongs to the aminoglycoside class of the antibiotics and also exhibits concentration-dependent bactericidal activity. Here it shows the image of the Micromonospora species under scanning electron microscopy alongside the microscopic appearance. In terms of the chemical properties, the gentamicin is a highly polar and water-soluble compound composed of multiple aminocyclitol rings. It has a molecular weight of approximately 477.6 grams per mole. It remains stable under the normal conditions such as exposure to light, air, and heat, which means that it does not require any special storage conditions. The figure shown here, it illustrates the chemical structures of gentamicin, which includes both the 2D skeletal in figure 2A, and also the 3D molecular structures in figure 2B. Moving on to its mechanisms of the actions, the gentamicin exerts its bactericidal effect by binding to the 30S ribosomal subunit, specifically at the 16 rRNA decoding site. These interactions, it can cause misreading of the mRNA during translation, which can produce the incorrect amino acids to incorporated into the growing protein chain. This could then lead to the production of defective or non-functional proteins. This disruption in protein synthesis then they would result in the bacterial cell death. Clinically, the gentamicin is considered a broad-spectrum antibiotic, particularly effective against aerobic gram-negative bacteria, which is commonly used to treat moderate to severe infections such as sepsis, urinary tract infections, pneumonia, and neonatal infections. It may also be used in combination therapy with other antibiotics, such as beta-lactams, to enhance effectiveness against certain gram-positive bacteria, like Staphylococcus aureus. Due to the poor absorptions of the gentamicins in gastrointestinal tract, these antibiotics cannot be administered orally, so instead they will be given via parenteral routes such as intravenous, intramuscular, and also subcutaneous administration sometimes. One of its advantage is that they are relatively low cost and also widely available, making it commonly used in hospital settings. However, the limitations is that the careful dosing and monitoring will be required because these antibiotics they can tend to lead to some side effects such as nephrotoxicity and also the ototoxicity, which they could affect the kidney damage and also they can cause hearing loss. And another issue is that the prolonged or inappropriate use of these antibiotics that can contribute to certain development of antibiotic resistance, and in this case I use Pseudomonas aeruginosa as an example. There are four main mechanisms by which the bacteria can develop resistance to gentamicin. The most common method is the enzymatic inactivation by aminoglycoside modifying enzymes that would chemically alter and inactivate the drug. Secondly, the reduced uptake can occur due to changes in the bacterial membrane, limiting the drug entry into the cell. Third, efflux pumps may actively remove the antibiotics from the bacterial cell, lowering its intracellular concentration. Lastly, target modifications through mutations in the 30S ribosomal subunit can reduce gentamicin binding. So this figure 5 shows the mechanism of the resistance. In conclusion, the gentamicin remains an important antibiotic in clinical practice due to its strong bactericidal activity against the serious infections. However, its use must be carefully controlled due to their potential toxicity and increasing issues of antimicrobial resistance globally. So for further information or references, you may scan the QR code at the bottom left corner of this poster. I will end my presentations here. Thank you very much for listening.